The two main types of tests that we use to determine MIC and MBC are going to be the disc diffusion methods and the broth dilution tests. The disc diffusion method, as you see right here, similar to the Kirby-Bauer test, can tell you the MIC, but it can't tell you if a drug is bactericidal or just bacteriostatic. Remember, bactericidal means it actually kills the pathogen, whereas if it's bacteriostatic, it's just going to inhibit replication. So that's where the broth dilution tests come in. So let's look specifically at broth dilution tests. After determining the minimum inhibitory concentration by making a sequence of decreasing concentrations of the drug in broth, that broth can then be inoculated with test bacteria. So basically they put the bacteria with the drug, right here, and then they inoculate it a new medium without the drug again, and if there is growth, then that means that the concentration of that drug was only bacteriostatic, because in the presence of it, it wouldn't grow, but then when that drug was gone, it would grow. However, if there isn't new growth, that means that the drug at this concentration actually killed it, and so then when it was incubated again without the drug, there was no growth because it was dead and it couldn't replicate. So if growth occurs on the plate, then the drug was just bacteriostatic. So let's look at these really quick and let's see if we can determine the MIC and the MBC. The minimum inhibitory concentration is going to be the lowest concentration that prevents growth. So if we were to look here, we have concentration, this lowest antibiotic concentration going to the highest concentration. We see that with 2 there is growth, but there's not growth with 10. So the minimum inhibitory concentration is going to be these 10 microliters per milliliter. The reason why is because when it is with the antibiotic, there is no growth. But let's see what the minimum bactericidal concentration is. Remember, if it's bactericidal, that means it's going to totally kill it. Right here at 10 microliters per milliliter, it simply inhibits the growth. But then when that antibiotic drug is gone, the bacteria grows again. So if we increase the concentration to 15 micrograms per milliliter, then we see that that is the minimum bactericidal concentration, to where it will not just stop growth, but it will actually kill the bacteria, to where when it is incubated in a subculture without the antibiotic, it will still not grow because it's dead. So on this, the MIC is going to be 10 and the MBC is going to be 15. Now let's look at something completely different, or as Monty Python would say, and now for something completely different. We're going to take a look at antibiotic resistance. The majority of available antimicrobial agents are effective against bacteria. We talked about how there are not as many antimicrobial agents out there for viruses and protozoans and helminths because it's more difficult to target them. However, let's see. So the majority of available agents are antibiotics. An interesting fact is that more than half of our antibiotics that are supposed to kill bacteria are actually produced by bacteria. Antibiotics, however, are not as useful as they once were, and that's because of antibiotic resistance. Antibiotic resistance can occur for a number of reasons. It can be due to just random mutations. It can be due to resistant genes on plasmids and then transposons that are transferred between bacteria. It can be due to bacterial conjugation. And it can happen in pretty much any type of bacteria. It's not restricted just to gram-positive or gram-negative or acid-fast or whatever. It happens in all kinds of bacteria. And there are 4 main mechanisms of antibiotic resistance, 4 main ways that bacteria can prevent antibiotics from really affecting them. That's enzymatic destruction of the drug, prevention of penetration of drug, alteration of drug's target sites, and rapid ejection of the drug. This slide shows you a picture that helps to explain each of those. First of all, you can have a bacteria that can block the entry of the antibiotic. If the antibiotic can't get into the cell to disrupt processes, then it's not going to have any effect. And you can take all the antibiotic you want, but it's not going to do any good because it can't get into the cell. Another thing it can do is it can inactivate enzymes. So the antibiotic does get into the cell, but the bacteria then can just make it not work. So it's inactive, the enzyme doesn't work anymore, and therefore there's no effect. The other thing it can do is it can alter its own target molecule that the antibiotic is against. So if you have an antibiotic that is against this target molecule right here, if it can change the shape of that, remember things are a lock and key fit, then this lock and the key will not fit the lock, and therefore there will be no result. And finally, some bacteria have developed resistance by being able to get rid of the bacteria or the antibiotic very quickly before it can have any effect. So the antibiotic gets in, but before it can have an enzymatic response or it can get to the target molecule or whatever, it is ejected from the bacteria right away.